Have you ever wondered why some people can drink milk their whole lives without, any problem, while others really struggle with it? Or maybe how a snake can literally rebuild its digestive system after a huge meal. I mean, basically tearing it down and then building it back up in just hours. Welcome to the deep dive. Today we're taking a really fascinating plunge into how animals, from the tiniest little organisms all the way up to the biggest whales, and yeah, even you and me, how we all get, process, and use our food. Our mission, as always, is to cut through the complexity, pull out the most important nuggets of knowledge from our source, which today is Animal Physiology, 4th edition, by Hill, Wise, and Anderson. We want to give you a shortcut to being genuinely well-informed about the incredible physiology behind it all. Get ready for some real aha moments, I think. Some surprising facts and hopefully a clear understanding of the different strategies animals use, why they're important for survival, their adaptive significance, and even some of the clever experiments scientists use to figure all this stuff out. So what does this all mean for you?
Well, you'll gain a richer appreciation for that intricate dance between an animal and its environment and how really every bite we take is part of this much grander biological story. Okay, let's jump right in. Maybe start with the fundamental question. Why do animals need to eat repeatedly throughout their lives, even after they're fully grown? It's not just about getting bigger, is it?
No, that's a great place to start. It's easy to think of an adult body as, well, kind of fixed. But the reality is your molecular self is in constant flux. Individual cells, molecules, they're always aging, getting damaged, being discarded and replaced. So there's this constant turnover. And that means some chemical building blocks are inevitably lost. They have to be replaced from food. And beyond that, think about energy. Every bit of chemical bond energy used in metabolism for moving, thinking, just staying warm, it can't be reused. It's gone. So new energy has to constantly come in through food.
Right. It's powerful to think of our bodies as being in this perpetual state of, renewal. Not static at all. And when we look at what makes up an animal body, setting aside water for a moment, we're talking proteins, lipids, minerals, nucleic acids, carbohydrates. But at the really basic atomic level, what are the most abundant elements?
Well, the big three are definitely carbon, hydrogen, and oxygen. They're just everywhere in living things. Nitrogen is also super abundant because it's in all proteins and nucleic acids. Then you have calcium, obviously crucial for skeletons, and phosphorus, which is in skeletons too, but also really important in cell membranes and energy molecules like ATP.
Okay, and among those building blocks, proteins seem to hold a really special place, almost like the foremost nutrient, as the name implies. About half the organic matter in mammals is protein. So what makes them so vital?
Proteins are just incredibly versatile. They're the ultimate multitaskers in the cell. They do so many things. Enzymes speed up biochemical reactions. Muscle proteins allow for movement. Structural proteins like collagen give shape and support. And then you've got receptors receiving signals, hormones sending messages, transport proteins like hemoglobin carrying oxygen. The list just goes on. Life, as we know it, wouldn't function without them.
And these amazing proteins are built from, 20 to 22 standard amino acids? But I understand there's a key challenge, particularly with nitrogen.
Absolutely. Proteins are roughly 16% nitrogen by weight, and nitrogen is often what we call a limiting element in ecosystems. This is because most organisms, animals, plants, most microbes can't just grab nitrogen gas, N2, out of the atmosphere. It's abundant, but unusable in that form. They need nitrogen that's already been fixed into chemical forms like nitrate or ammonium, and those are often in short supply. So this nitrogen limitation can ripple right up the food chain, affects herbivores than the carnivores that eat them.
Huh. So even if an animal eats a lot, if that food lacks enough nitrogen or the right kind of nitrogen building blocks amino acids, it's a problem. And animals can't make about 10 of these standard amino acids fast enough themselves. That makes them essential, meaning they have to get them from food.
Exactly. They're essential dietary requirements. And the specific list can vary a bit between species or even between, say, a growing animal and an adult. A growing rat needs 10, adult humans typically 8 or 9. But the core idea is the same. If you can't make it or can't make enough of it, you absolutely must eat it.
Okay, help me understand this. Why don't animals just store amino acids? Like they store fat. Seems like it would make things easier having a reserve.
Yeah, it's one of those slightly counterintuitive things. But animals generally do not store amino acids for later use in protein building. When you inject excess amino acids, the nitrogen part is quickly stripped off and usually excreted as waste, like urea. The leftover carbon chains are then used for energy or maybe converted into fat or carbs. It's very much a just-in-time strategy.
Like modern manufacturing, almost.
Kinda, yeah. The amino acid building blocks have to arrive pretty much exactly when the protein synthesis machinery needs them.
And the consequence of that sounds pretty significant. What happens if even one of those essential amino acids is missing from a meal?
It really limits things. If you're short on, say, lysine, even if you have tons of all the other essential amino acids, you can't build proteins efficiently that require lysine. So those other abundant amino acids effectively get wasted, at least for protein building. They'll be deaminated and used for energy instead. Lysine deficiency, for instance, is a serious health issue for millions globally.
Wow. So the solution for many animals, including us, is often mixing foods, like the classic corn and beans combo or milk and cereal, to get a more complete set of amino acids.
Precisely. Different foods often have complementary amino acid profiles. Mixing them helps ensure you get all the essential ones together at the same time, increasing the overall protein utilization. And ultimately, for most animals, the original source of all these essential amino acids is plants and algae. They're the ones that can synthesize all 2022 from scratch.
Okay, let's shift gears a bit. Let's talk lipids, or fats. Often just as abundant as proteins. We're talking nonpolar, water-hating molecules, mostly carbon and hydrogen. And they contain fatty acids. What are their main jobs in the body?
Lipids have several key roles. Phospholipids and cholesterol are fundamental components of every cell membrane. They form that essential barrier. They're also fantastic energy stores. Gram for gram, they pack way more energy than proteins or carbohydrates. For animals on land, lipids in the skin are also crucial for waterproofing, reducing water loss. And certain lipids, the steroid lipids, function as really important hormones, regulating all sorts of body processes.
So nutritionally, are lipids generally less problematic than proteins? Easier to manage?
In some ways, yes. Animals are generally quite good at synthesizing many types of lipids from other molecules, like carbs or proteins. Plus, and this is a key difference from proteins, animals do store lipids. Excess fat is stored for future use. That's why we can, put on weight.
But there's still essential fatty acids, aren't there? Like the essential amino acids, ones we have to get from our diet.
That's right. Problems can definitely arise there. Many animals, mammals included, lack the enzymes to create certain types of double bonds, specifically at the omega-3 and omega-6 positions in the fatty acid chain. So fatty acids like alpha-linolenic acid and omega-3 and linoleic acid and omega-6 are essential. Got to get them from food. And some animals, like cats or certain fish, have even more specific fatty acid requirements.
Okay, what about carbohydrates? Let's see, maybe a bit simpler. Monosaccharides like glucose, disaccharides like sucrose, and the big polysaccharides like starch and glycogen. What are their main roles?
Carbs mainly do three things: structural support, energy storage, and energy transport. Structurally, you've got large polysaccharides like chitin, the stuff arthropod exoskeletons are made of, and cellulose in plants. It's actually kind of amazing. Chitin and cellulose are the two most abundant organic compounds in the entire biosphere.
Wow, I didn't know that. Chitin and cellulose.
Yeah. Then for energy storage, you have starch in plants and glycogen in animals.
Right, glycogen. How does that compare to fat for storage?
Well, glycogen is stored with a lot of water. It's highly hydrated, so it yields less energy per unit of weight compared to lipids. It's more of a readily accessible short-term glucose store. Really important for the brain and vertebrates, which runs primarily on glucose, and for quick bursts of energy in muscles. And for transport, small sugars like glucose in our blood or trehalose in insects are dissolved in body fluids to shuttle energy around.
And are there big nutritional challenges with carbs? We mentioned cellulose being hard to digest.
Yeah, that's the main one. While there aren't really essential carbohydrates that animals can't make, the indigestibility of structural ones like cellulose and chitin is a major issue for many animals. Humans, for instance, lack the enzyme cellulase, so plant fiber just passes right through us. We can't get energy from it directly.
Okay, let's move to the micronutrients, vitamins and minerals. Needed in small amounts, but they have huge impacts. First, vitamins. Essential organic compounds can't be synthesized by the animal. How did we even figure out they existed?
It really started with experiments, like feeding mice highly purified diets. Scientists found the mice couldn't grow properly unless these trace accessory food factors, as they were first called, were added back.
That's where it gets really interesting, the evolution of why these things are essential in the first place.
Exactly. It raises that question, why can't animals just make them? And often the answer seems to be evolutionary opportunism, or you could say evolutionary shortcuts. Take vitamin A, retinol. Animals can't synthesize its core structure from scratch. Instead, during evolution, they co-opted this structure, which was already being made by plants and algae in the form of carotenoids. They incorporated it into crucial molecules like rhodopsins for vision. It was easier to just use what was available than to evolve a whole new complex synthesis pathway.
So it's a story of using pre-existing building blocks rather than reinventing the wheel.
Precisely. But the consequence is dependency. If the vitamin isn't in the diet, you have a problem. Vitamins are chemically very diverse, but functionally they often get incorporated as key parts of larger molecules, frequently acting as cofactors that help enzymes do their jobs. We usually group them into water-soluble, like the B vitamins, which are vital for basic metabolism in pretty much all animals, and vitamin C and lipid-soluble A, D, E, and K, which vertebrates need for more specialized things like vision, calcium balance, antioxidant defense, and blood clotting.
Okay, and then there are minerals, essential chemical elements beyond the big four: carbon, hydrogen, oxygen, nitrogen. We need over 20 others. What are they doing?
They have incredibly diverse roles, too. Many enzymes require metal ions, iron, copper, zinc, magnesium to function properly. They're called metalloproteins. Iodine is essential for thyroid hormones. Phosphorus, as we mentioned, is critical for cell membranes, DNA, RNA, and bone. Sodium, chlorine, potassium are the main ions determining the properties of our body fluids.
It's wild to think of the human body as this complex chemical formula with billions upon billions of atoms of hydrogen, oxygen, carbon, nitrogen, and then smaller but absolutely critical amounts of calcium, phosphorus, potassium, sulfur, sodium, chlorine, magnesium, iron, zinc, iodine.
It really puts it in perspective, but mineral deficiencies are a huge problem globally, especially for land animals. Soils in many regions can be poor in essential minerals like sodium, phosphorus, calcium, magnesium, copper. This affects livestock and wildlife. An iodine deficiency, for example, affects well over 1 billion people. It's a leading preventable cause of mental retardation, but easily fixed with iodized salt.
So what does this all mean for, say, that huge migration of wildebeest in the Serengeti? Is that driven by minerals?
If we connect this to the bigger picture, yes, that's a leading hypothesis. The migration seems to be, at least in part, a quest for minerals. The soils in the southeast Serengeti are richer in minerals because they're from more recent volcanic activity. Grasses there can have, say, 1.5 times more calcium. Timing their calving season to be in those mineral-rich areas is likely a crucial strategy to ensure the young get enough minerals for healthy growth and development. It's a massive ecological phenomenon driven by basic chemical needs.
A truly grand biological story indeed. Okay, so animals have these fundamental needs. Now, how do they actually get the food? It's not just random eating, right?
Yeah.
Animals often select foods to meet specific needs.
Definitely. Behavioral selection is key. We see mountain gorillas seeking out rotting wood specifically for its sodium content, or experiments showing wolf spiders choosing flies richer in protein or lipids depending on what they need to balance their diet. They're making sophisticated choices.
And beyond choosing what to eat, there's an incredible diversity in how they eat, the mechanisms themselves.
An absolutely stunning array. One major strategy is attacking individual prey items. Think orcas hunting seals, fish swallowing other fish whole, sea stars averting their stomachs onto prey.
Butterflies sipping nectar with a proboscis, falcons striking birds in midair.
Exactly. And even within related groups using similar tools, you see amazing specialization. Look at bird bills, the woodpecker's chisel, the cardinal's seed cracker, the sandpiper's delicate probe. Or think about grazers like zebras and wildebeests on the African plains. Slight differences in their teeth, lips, and muzzle shape allow zebras to eat taller, stemmier grasses while wildebeests focus on shorter leaves. This helps them coexist.
And snails. You mentioned them having a radula, like a rasping tongue. But that basic tool evolved into some pretty extreme forms.
Oh, absolutely. The primitive form scrapes algae, but then you get carnivorous drill snails that use the radula to bore precise holes through shellfish shells. And the most spectacular are the cone snails. Their radula teeth have become detachable, barbed harpoons connected to a venom gland. They inject potent neurotoxins, conotoxins, to instantly paralyze fish. It's incredible weaponry.
Speaking of toxins, they seem to be a recurring theme used by predators, but also by prey for defense.
It's a chemical arms race. Predators like snakes, spiders, jellyfish use venoms, often protein-based, to subdue prey. Prey can have defensive venoms too, like bees or wasps, but more common, especially in sessile things like plants, sponges, or seaweeds, are toxic or repellent secondary compounds laced throughout their tissues to deter being eaten. And then some herbivores or predators evolve ways to detoxify or tolerate these chemicals, specializing on otherwise poisonous food sources. It's constant biochemical warfare.
Okay, a totally different approach is suspension feeding. Eating tiny things suspended in water plankton, detritus collected in bulk, not individually targeted, like filker feeding clams or baleen whales eating krill.
Right. This is super common in aquatic environments because there's just so much microscopic food available. A huge advantage is that it lets animals feed lower on the food chain, tapping into a much larger energy base.
That's the 10% rule connection, isn't it? Explains why so many huge animals are suspension feeders.
Exactly. If we connect this to the bigger picture, remember, only about 10% of energy makes it from one trophic level to the next. So a whale eating krill, which eat phytoplankton, gets maybe 100 times more energy from the same amount of primary production than a whale eating large fish that ate smaller fish that ate zooplankton that ate phytoplankton. Shortening that chain makes a massive difference. That's why blue whales, whale sharks, basking sharks, huge schools of herring, even krill themselves, are suspension feeders. They're tapping into that vast base of the food web.
But the challenge is gathering enormous numbers of tiny particles. How do they do it? Baleen whales use those billion plates.
Yes, keratin plates hanging from the upper jaw acting like a sieve. But for many suspension-feeding fish, it's not just simple sieving with their gill rakers. Recent work shows it's more complex hydrodynamics. Water flows past the rakers in a way that concentrates particles near the mouth for swallowing, while letting relatively clean water escape through the raker slits. Pretty sophisticated fluid mechanics.
Incredible ingenuity. But some animals take it even further, outsourcing food production to microbes living inside them, like internal gardeners.
A key insight here is how some animals let microbes do the primary production for them. Some aquatic animals host photosynthetic algae internally. The algae capture sunlight, make sugars, and leak some to the host, sometimes meeting 100% of the host's energy needs.
Reef-building corals being the classic example. They're basically animal-algae partnerships.
Absolutely. Those zooxanthella algae living inside coral tissue are essential. Coral bleaching happens when the algae leave, often due to stress like warm water, and the coral usually dies without them. It also explains why reefs need clear, sunlit water. If the water gets too murky, the algae can't photosynthesize, and the whole system collapses.
Then there are the chemosynthetic guys, the hydrothermal vent communities discovered in the deep sea. That was a mind-blowing discovery, right? Life thriving in total darkness based on chemical energy.
Completely changed our understanding of where life could exist. The key players are chemosynthetic bacteria, particularly sulfur oxidizers. They use the chemical energy released from oxidizing hydrogen sulfide, that rotten egg smell spewing from the vents, to build organic matter. Sulfide comes from seawater reacting with hot rocks below. The most dramatic example is the giant tube worm, Riftia. Grows up to 1.5 meters long but has no mouth, no gut, no anus. It's packed with these sulfur-oxidizing bacteria in an organ called a trophosome. The worm's specialized hemoglobin binds both oxygen and sulfide from the water and delivers them to the bacteria, which then feed the worm. It's an amazing symbiosis.
Just incredible. And finally, there are the heterotrophic microbes, the ones that break down organic matter, living in animal guts, the gut microbiome. We all have one.
Yep, humans, probably all animals. Colonized from birth, trillions of microbes actually outnumbering our own cells by maybe 10 to 1, and we're increasingly realizing how much this gut microbiome influences our physiology, affecting metabolism, immunity, even risk of diseases like obesity or diabetes. Different people can have different enterotypes, different microbial communities that might be better or worse at certain things, like making vitamins or breaking down complex carbs.
And beyond the general microbiome, there are those highly specialized symbioses, particularly in herbivores, involving fermenting microbes in enlarged gut chambers, like the rumen or cecum. So what's the big advantage here? What superpower do these fermentation chambers give the animal?
If we connect this to the bigger picture, these microbes offer at least 3 massive nutritional benefits. First, they break down stuff the animal can't digest on its own, especially cellulose. They ferment it into short-chain fatty acids, or SCFAs, like acetic, propionic, butyric acid. The animal can absorb and use these SCFAs for energy. This process also makes gas, CO2, and methane, which ruminants famously belch out. Second, these microbes synthesize essential nutrients. Rumen microbes make all the B vitamins and all essential amino acids. The host animal then gets these nutrients by simply digesting the microbes themselves as they pass further down the gut. And third, they allow for nitrogen recycling. Waste nitrogen, like urea from the host's metabolism, can diffuse into the rumen. Microbes break it down, use the nitrogen to build their own proteins. When the host digests the microbes, it reclaims that nitrogen. It's a brilliant way to conserve a limiting nutrient.
A total game changer for herbivores, letting them live on plant fiber. We see this in foregut fermenters like cows, sheep, deer, beer, the ruminants with that big rumen chamber at the start of the stomach.
Right. And also kangaroos, hippos, even the Hoatzin bird. The rumen acts like a big fermentation vat. Then you have hindgut fermenters, horses, rabbits, elephants, apes, some birds, reptiles, fish. They have enlarged chambers in their large intestine or cecum further down the line.
What's the difference for them?
They still get the SCFAs absorbed, but any vitamins or essential amino acids made by the microbes in the hindgut tend to get pooped out because they're past the main absorption site of the small intestine. So many hindgut fermenters, like rabbits, practice coprophagy. They eat their own feces to recover those microbial nutrients. A bit gross, maybe, but effective.
Nature finds a way.
Yeah.
And invertebrates have these partnerships too.
Oh, yes. Termites are famous for it. They have protists and bacteria in their hindgut to break down cellulose and wood. Many beetles, too. These symbionts often provide vitamins and amino acids, recycle nitrogen, and sometimes do even more specialized things, like synthesizing sterols for insects or helping blood feeders like leeches digest blood and even producing antibiotics.
Okay, so the food is acquired, maybe with some microbial help. Now let's get into digestion and absorption, breaking food down and getting it into the body. Digestion uses enzymes. Absorption gets the molecules into the tissues.
That's the basic flow. In vertebrates and arthropods, digestion in the gut lumen generally comes first, then absorption. But in simpler animals like sponges or bivalves like clams, it's often the other way around. Cells absorb small food particles first and then digest them intracellularly.
The vertebrate gut usually has that four-part structure: head gut, foregut, midgut, hindgut.
Right. Head gut for capture and initial processing. Foregut, esophagus, stomach, for transport, storage, and starting protein digestion with acid and pepsins. Midgut, small intestine, is the main event. Most digestion of carbs, fats, proteins happens here, and most absorption of nutrients, water, vitamins, minerals. Receives key juices from the pancreas and liver. And the hindgut, large intestine, is mainly for water and mineral reabsorption, compacting waste. Super important for hydration. Cholera kills by disrupting hindgut water absorption.
And muscles move it all along peristalsis, pushing forward, segmentation mixing things up. Controlled by that second brain, the enteric nervous system. How do insects or clams compare?
Arthropods, like insects, are quite similar to vertebrates, mostly extracellular digestion, muscular movement. Bivalves are different, much more intracellular digestion, and they use cilia, tiny hairs, to move food particles around internally.
Digestive enzymes are incredibly specific, aren't they? Each one targets a specific chemical bond, like chitinase for kinin, which we lack.
Exactly. Highly specific hydrolytic enzymes, usually. And they operate in different locations. Some are intraluminal, secreted right into the gut cavity to mix with food. Some are membrane-associated, embedded in the gut lining cells, acting on food that contacts the cell surface. And some are intracellular, working inside the cells after particles have been absorbed. Most animals use a combination.
For carbs, the enzymes breaking down disaccharides like sucrose or lactose are often membrane-associated in the midgut. And polysaccharides need a couple of steps.
Usually, yeah. An enzyme like amylase in saliva and pancreatic juice breaks down starch into smaller sugars first. Then other enzymes like maltase or sucrase, often membrane-bound, break those down into simple monosaccharides like glucose, which can be absorbed. And again, digesting cellulose or chitin requires specialized enzymes that many animals, including vertebrates, just don't have.
Protein digestion seems more complex, lots of different bonds to break, and the body has to avoid digesting itself.
It is intricate. It usually involves endopeptidases, cutting inside the protein chain, and exopeptidases, nipping amino acids off the ends. And yes, self-protection is critical. These powerful enzymes are made as inactive precursors, zymogens, and only activated where and when needed. In vertebrates, it starts in the stomach with pepsins activated by acid. Then, in the midgut, a whole suite of pancreatic enzymes, trypsin, chymotrypsin, etc., also secreted as zymogens, get activated and continue the breakdown. This yields free amino acids and short peptides. Those peptides are then broken down further by peptidases on the midgut cell surface, and some small and tripeptides are even absorbed into the cells and broken down there by intracellular peptidases. The final product going to the blood is mostly free amino acids.
And lipids. Simpler enzymes, but the challenge is they don't mix with water. Emulsification is key.
Exactly. You need to break up large fat globules into tiny droplets to increase the surface area for lipases to work. In the vertebrate midgut, pancreatic lipases do the digesting, but the crucial step is emulsification by bile salts from the liver. They act like detergents. Bile salts get recycled too, absorbed further down and reused. The final absorbable products are mainly free fatty acids and monoglycerides.
Okay, so everything's broken down. How do these molecules actually get into the body tissues? The gut lining is often heavily folded with villi to massively increase surface area, right?
That huge surface area is vital for efficient absorption, and transport across the gut lining uses different mechanisms. Simple diffusion, facilitated diffusion using protein channels or carriers, and active transport, which requires energy, often using ion gradients like sodium. Water-soluble molecules, sugars, amino acids, water-soluble vitamins generally need transporter proteins to help them cross cell membranes.
So how does your body get that sugar, like glucose, from your breakfast cereal into your bloodstream? It sounds simple, but the cell biology is pretty involved.
This raises that important question of cellular transport, yeah? Glucose uses a specific transporter called SGLT1 to get into the midgut cells from the gut lumen. This is secondary active transport. It actually uses the energy stored in the sodium gradient across the membrane. Then, to get out of the cell and into the blood, glucose uses a different transporter, GLUT2, which works by facilitated diffusion. Fructose uses different transporters, GLUT5 to get in and GLUT2 to get out. Amino acids are even more complex, with multiple different transporters, mostly using active transport linked to sodium or other ions. Specific active transporters also exist for some B vitamins and vitamin C.
What about the fatty stuff? Fatty acids, monoglycerides?
Being lipid-soluble, they can diffuse across cell membranes more easily, but they aren't very soluble in the watery gut contents. That's where bile salts come in again. They form tiny structures called micelles around the fatty acids and monoglycerides, keeping them dissolved and bringing them close to the cell surface. The lipids then dissociate from the micelle and diffuse into the cell. Inside, they're reassembled into triglycerides, packaged with proteins into particles called chylomicrons, and exported into the lymph system, eventually reaching the blood. Short-chain fatty acids are a bit different. They're absorbed more directly, often by diffusion, wherever they're produced.
It's clear digestion isn't static. The system responds dynamically, both immediately after a meal and over longer timescales. Let's talk about those acute, short-term responses to eating.
Yeah, it's a highly coordinated response involving nerves, especially the enteric nervous system and hormones. Swallowing triggers peristalsis in the esophagus. The stomach relaxes to receive food. Food presence stimulates G cells to release the hormone gastrin. Gastrin then tells other stomach cells to secrete acid and pepsinogen, the precursor to pepsin, and stimulates stomach muscle contractions to mix everything up.
And the stomach doesn't just dump everything into the midgut at once, right? It meters it out.
Exactly. The pyloric sphincter controls the flow into the small intestine. As acidic, partly digested food enters the midgut, it triggers the release of other hormones like secretin, CCK, and GIP. These hormones do several things: stimulate the pancreas to release digestive enzymes and bicarbonate to neutralize the acid, stimulate bile release from the liver and gallbladder, and importantly, they also act back on the stomach to slow down its emptying and acid secretion. It's a feedback loop saying, hey, slow down, the midgut is busy.
It's like sophisticated traffic control.
Very much so. And it's not just vertebrates. Even something like an oyster shows dynamic changes, with digestive cells rapidly changing structure and function in response to feeding.
What about controlling hunger and feeling full? Satiation. That seems incredibly complex, involving the brain.
Hugely complex. It involves coordination between the gut, brain, especially the hypothalamus and hindbrain, pancreas, and even fat tissue. Ghrelin, mainly from the stomach, tends to rise before meals, stimulating hunger. Satiation, feeling full and stopping eating, involves a whole host of signals. Stomach stretch sends quantity signals. Hormones released from the intestine and pancreas in response to nutrients like CCK, PYY, GLP-1 send composition signals. And leptin from fat cells provides a longer-term signal about energy stores, generally inhibiting appetite. It's a multi-layered system.
Okay, zooming out again. Longer time frames. Many animals naturally fast for long periods, bears hibernating, penguins incubating eggs, salmon migrating. This isn't starvation, right?
No, natural fasting is physiologically distinct from starvation. The animal maintains well-being, often relying on stored reserves in a controlled way. Starvation implies deterioration. Pacific salmon, polar bears, emperor penguins, they all undergo remarkable programmed fasts.
Okay, let's unpack this. Pythons, they can go weeks between meals. How on earth do they manage that incredible transition from fasting to digesting a massive meal? How do they bounce back?
It is genuinely one of the most extreme examples of physiological flexibility known. It's where it gets really interesting. Between meals, pythons dramatically downregulate and effectively partially deconstruct their digestive tract to conserve energy. The gut shrinks. But within 24 to 48 hours of swallowing prey, they undergo explosive reconstruction. The midgut mass can double. Transporter proteins for glucose and amino acids increase maybe 20-fold. It's driven by massive coordinated changes in gene expression. Thousands of genes are turned up or down almost immediately after feeding. Their metabolic rate skyrockets, maybe 40-fold. The heart muscle even grows larger temporarily to support it all. Stunning adaptation.
Truly stunning. That's an extreme case. But animals also adapt their digestion to more gradual, chronic changes, like shifts in diet.
Absolutely. If an animal's diet changes consistently, its digestive system remodels over days or weeks. It upregulates the specific enzymes and transporters needed for the new food type. Eat more carbs, you make more carb-digesting enzymes and glucose transporters. Eat more protein, you boost protease activity. Gut size itself can also adapt. Small birds and mammals might increase gut length in the cold to process more food for heat generation. Pythons are the ultimate example of size change, and dietary components themselves can directly influence gene expression related to digestion and metabolism. Fatty acids, for instance, can bind to receptors inside cells and regulate genes involved in handling fats.
Which leads us to evolutionary changes over generations. Populations adapting their digestive physiology.
Yeah.
So what does this all mean for milk drinking? That difference between people who can digest lactose as adults and those who can't. What's the evolutionary story there?
If we connect this to the bigger picture, yeah, lactase persistence, the ability to digest milk sugar as an adult, is a classic example of recent human evolution. It evolved independently in several different populations that adopted dairy farming, like in Northern Europe and parts of East Africa. Different genetic mutations arose, but they all had the same effect, keeping the lactase enzyme active into adulthood. These mutations spread rapidly within just a few thousand years, because being able to get nutrition from milk provided a huge survival and reproductive advantage in those cultures. It really highlights how populations can diverge physiologically based on diet and lifestyle.
And this parallel evolution of digestion and diet happens across the animal kingdom.
Yes, you see correlations. Animals eating lots of nectar tend to have high levels of sucrose, the enzyme that digests sucrose. Insectivores often have high trihylase activity for digesting insect sugar. Herbivores generally show higher capacity for glucose absorption than carnivores, reflecting their different dietary inputs, and some of this seems genetically ingrained.
Finally, digestion also changes predictably within an individual's life through development or internal clocks.
Right. Developmentally, the most obvious example in mammals is lactase production dropping off after weaning. Other enzymes for solid food might increase then. Frog metamorphosis is another dramatic one. The gut completely restructures as the aquatic herbivorous tadpole changes into a terrestrial carnivorous frog. And then there are clock-driven rhythms.
Like daily circadian rhythms,
Yeah.
affecting digestion even if you're not eating.
Exactly. Nocturnal animals like rats show daily cycles in their digestive enzyme activity, peaking during their active nighttime feeding period. It anticipates the need. And there are longer circannual rhythms too, like hibernators automatically fattening up in autumn, preparing for winter, even if kept in constant lab conditions. These cycles are driven by internal biological clocks, preparing the animal for predictable seasonal changes.
Wow. We've covered a huge amount, from the fundamental need for nutrients, the incredible variety of feeding strategies, the complex chemistry of digestion and absorption, right through to these amazing dynamic responses and adaptations over time. We really hope this deep dive has given you a fresh perspective on these absolutely fundamental processes that sustain all animal life, including, of course, your own. It's just an amazing testament to evolution and the intricate coordination happening inside every living thing. So here's a final thought to leave you with. Given the incredible plasticity we've seen pythons rebuilding guts, human populations evolving lactase persistence relatively quickly, what kinds of dietary challenges might future human generations face? And how might our own physiology continue to adapt and evolve in response? Something to ponder. Thank you so much for being part of the Last Minute Lecture family.